Okay, now let us look at the classification of the enzyme. Okay, enzyme is classified according to the International Unions of the Biochemistry and Molecular Biology. All the enzyme, when we want to do the classification, bear in mind, we classify it according to the role of the enzyme. So based on 6 different potential chemical reactions that can happen in the biological process, so there will be 6 major class of enzyme. So before this, let us look at the naming of the enzyme. In order to name the enzyme, normally we name the enzyme according to the substrate, okay, according to the substrate. And also the enzyme must have the ending with the suffix ase, ase, okay, suffix ase. And nowadays, even they have some of the enzyme which is not named according to the substrate, they also have, but most of the time we are naming the enzyme according to substrate. So there are 6 class of the enzyme, which is the hydrolase, isomerase, transferase, lyase, oxidoreductase, and ligase. Altogether, there will be 6 of them. Okay, so hydrolase is actually a class of enzyme that speed up the hydrolysis reaction. So when we're talking about hydrolysis reaction, it is actually a process to break down the covalent bonds by adding the water. So we have a lot of enzyme that found in the digestive system, which is category under the hydrolase. They are maltase, which is break down the maltose. And then we have the sucrase that can break down the sucrose. And then we have the amylase that can break down the carbohydrates or the starch. So we have this kind of enzyme, the example of enzyme, which is category in hydrolase. Isomerase is actually a class of the enzyme that catalyze the rearrangements of the molecule structures. Okay, the rearrangement of the molecule structures. They can rearrange the molecule structures and changing the functional groups of the molecule, but most of the time, the end product must be an isomer. Okay, must be the isomer for the substrate. Okay, the process that involved is called isomerizations. Okay, and isomerase, we have the isomerase and also the mutase. Okay, the mutase, they are also found in category of the isomerase. So another examples of the isomerase, we have the phosphoglucomutase, so which will eventually transferring or rearrange the glucose 6-phosphate to the fructose 6-phosphate. This is happened in chapter 5. Okay, chapter 5, we're going to see this particular enzyme again. Transferase, which is, if you look at the explanation, it's like very similar to isomerase, but they are different. Transferase function is to transfer functional group from one molecule to another molecule. So they must be actually transferring the functional group from one molecule to another molecule. So if let's say I have the molecule A with the hydroxyl group, it's actually react with the B, and then at the end, the hydroxyl group is actually transferred to the molecule at B. So this particular reaction will require transferase. And what is the example of the enzyme is the hexokinase, which is again, this is the enzyme that you're going to learn in your chapter 5 later on in the first reaction, which is the glycolysis. Okay, lyase. Lyase is actually the enzyme which speed up the breakdown, or they are actually carry out lysis, well, which is the breakdown of the chemical bond from a complex molecule into simple molecule. But different with hydrolase, they do not require water. They just simply break down the chemical bond. Okay, how to break down the chemical bond? By adding or removing different functional group. Okay, or small molecule like the carbon dioxide. They doesn't actually need the water to initiate the reaction, not like the hydrolase. Hydrolase required water. This one, no need. Okay, the process that they carry out is lysis. Oxidoreductase is the enzyme that carry out the redox reaction, meaning that they involve in the transfer of the electron. Okay, the transferring of the electron from one molecule to another molecule. Okay, so they catalyze the reduction and the oxidation. So we have the example like dihydrogenase, like the examples in the chapter 5 as well, the NADH dehydrogenase. They are oxidoreductase. And the last one is the category of ligase. This is a group of enzyme that catalyze or speed up the formation of the chemical groups. Okay, like the synthetase. Okay, we have the synthase, we have the synthetase. They are all ligase. Okay, they require the ATP in order to carry out the reaction. And the ligation is actually the process that they carry out. Ligation is the process that they carry out. They will have the tendency to actually forming the new chemical bond, and the chemical bond formation require the energy that's supported by the ATP. So that is the sixth class of the enzyme.